Great, thank you very much indeed. So it's the time to present the 25th anniversary MRC Millennium Medal. So this award was established in the year 2000 and it's been given 13 times to MRC-funded scientists who've been exceptional. Exceptional in delivering science, exceptional in having impact, and also influencing the wider research environment. I'm pleased to see some of the previous recipients in the audience today. The medal has been awarded to people who've advanced our understanding of epidemiology, diagnostics and biotechnology, chronic diseases, metabolic diseases, cardiovascular and neurodegenerative disorders, to name but a few. And all of the scientists are world leaders when they receive this award. And many of them actually go on to receive higher honours, either nationally or, in some instances, the Nobel Prize. So it's a highly prestigious award that the MRC is very proud of, and we have it made every time personally and personalised by the Royal Mint. So the winner is judged on a number of criteria that include scientific excellence and an international reputation, evidence of scientific impact on health, a commitment to improving the wider research environment, and also advancing the MRC mission to drive health improvements forward through world-class medical research. And the MRC is absolutely delighted to award the 25th anniversary Millennium Medal to Professor George Davey Smith, who is Professor of Clinical Epidemiology at Bristol Medical School and Director of the MRC Integrative Epidemiology Unit at Bristol University. This is a recognition for his pioneering MRC-funded research into causal influence, Mendelian randomization, and the impact that this has had on public health and medical research worldwide. George is one of the most respected and influential figures in modern epidemiology. He's known for finding better ways to understand what really causes diseases in populations and, critically, how they can be prevented. He's recognised for pioneering the technique Mendelian randomization, which you'll hear a little bit about shortly. This uses genetic information to work out whether certain factors such as diet, lifestyle, or blood biomarkers actually cause a disease rather than simply being associated with it. So it really gets towards the MRC's ambition to develop our mechanistic understanding of diseases so that we can diagnose them earlier, treat them better, and to prevent them in the long term. So Professor Davey Smith has made major contributions to understanding health inequalities through his work, and how health is shaped right across our life course. He's an instigator of several cohort studies which are internationally renowned, particularly noted for his work nurturing the Avon longitudinal study of parents and children, the ALSPAC study, which has led to pivotal discoveries on how social and behavioural and biological factors interact to influence health from prenatally through childhood and right into adult life and old age. He's championed the idea of triangulation, which is to use different types of evidence to build a clearer picture of cause and effect. And he's been a strong advocate for making research data open and accessible to the wider scientific community worldwide. So I would now like to invite Professor George Davey Smith to the stage to present him with the Millennium Medal and offer his congratulations from all of us at the MRC. George.
Yeah. This side.
I'll fall off. Not before you give me a lecture, please. Great. Over to you, George.
So it's a great honour to have received this medal. It'll be an even greater honour when my PowerPoints are up, because it is up. As an epidemiologist, I can't speak unless I've got a PowerPoint behind me. I'm going to talk about an MRC-linked career in epidemiology, and in particular about causal inference. I started out in epidemiology in South Wales in 1984. The man who looks like me is my dad, was my dad. I was working for a health promotion group called Heartbeat Wales. Never work for anywhere where they make you wear dodgy T-shirts. That's my one bit of advice. And I organised a survey around Wales, a clinical survey, where for a year we went round in an ambulance around all villages, towns in Wales, taking blood samples, measuring objective fitness measures, the first large-scale study in the UK with objective fitness measures, blood pressure, etc., on people as a baseline survey. But I was lucky enough to be, as I say, in Cardiff, and there was the MRC South Wales unit there with the wonderful 95-year-old and thriving Peter Elwood, who was director of the unit. And they had just set up the Caerphilly heart disease study. This was a small-ish, for now these days, cohort study, which was focused on looking at new risk factors for coronary heart disease, in particular higher HDL cholesterol, which was supposed to be protective, fibrinogen and other clotting factors, which were supposed to be adverse. And they were setting this study up, and that allowed me to go and spend time with this running study to actually learn how to do survey work, which was fantastic. That unit had been under the directorship of Archie Cochrane, who's on the right here. He's known for the Cochrane collaboration, which was set up quite a few years after Archie had died. He would be sitting in the top attic floor, smoking and doing the Times crossword. The only Cochrane collaboration in those days was helping Archie with the Times crossword, but it was a privilege to be in that environment. And so that MRC unit truly gave me a start in epidemiology. And in 1986, I was lucky enough to get selected for the 10-day teaching seminars. These are a series that have been going on since 1968 in cardiovascular epidemiology, where I managed to get to know Geoffrey Rose better. Geoffrey was actually on the oversight committee of Heartbeat Wales, but I hardly saw him because the time he turned up for a whole day visit. I'd been headbutted the night before when I was retrieving my girlfriend's cigarettes from someone who had nicked them in a pub, and it spectacularly spat in my nose. I had two big black eyes, and it was thought that wasn't the image that Heartbeat Wales was trying to, even though, of course, I wore the T-shirt, but that wasn't the image they were trying to convey. But then I got to know Geoffrey in Cuba, which was an amazing privilege, but also met a guy called Peter Sandiford, and through him I started working in Nicaragua with Matthias Egger in the back, who's laughing as I mention that name, and Jim Neaton, where we went and spent a lot of time in Minneapolis, who taught me a huge amount of epidemiology. Really pivotal point of my career. But the grant money ran out, or Heartbeat Wales funding ran out, and I was going to become unemployed. So Peter Elwood didn't have any MRC money, but he put in for a Welsh Office grant to study lead and blood pressure, which would support me. And I thought, well, I believe in chance, as you'll discover as I carry on talking. I thought, I'll be unemployed for a bit, and then if this grant comes off, I'll then stay here and do this work. And if the grant didn't come off, I was going to go to Montreal, to McGill, and do a PhD in neuropsychology with Brenda Milner. But the grant was funded, so neuropsychology's gain was epidemiology's loss, and I actually stayed in epidemiology because of that grant, and that grant was based at the MRC unit. And that allowed me to co-author, but I didn't write the first author, a series of very boring papers, evaluation of a automated blood pressure machine, and papers on the lead, null study. But then my first author paper was when I was 30, and by then I'd done the MSc in epidemiology at the London School of Hygiene part-time over 2 years. And through that and through Geoffrey Rose's influence, he had set up the Whitehall 1 study in 1967. I looked at blood cholesterol and primary brain tumours because there was some work on gliomas and cholesterol. And we showed an association, positive association. It makes me squirm to look at this paper because, as people who know me know, I hate the word significant or using those thresholds. But here it was, things were not significantly, or were, p less than 0.05, significantly related. So Jim Neaton had a massive cohort in Minneapolis. So we thought, well, we'll replicate this. So we didn't replicate this. In a much bigger study, there was a null relationship. Now, the BMJ had published the first one, and I went back to them and said, look, this paper is massively more powered. The BMJ obviously weren't interested, and we couldn't get it published for quite a lot of years. It went down the publication trail. But then in 1989, in Nicaragua, on the road to Granada, I had a rather serious motorbike smash. This is the car I went into. And that meant I wasn't very mobile. So I started writing. And so this is after, as I say, my first author paper was at 30, which is quite late these days. And I was particularly interested in health inequalities and wrote a substantial amount on that. And the Whitehall study had become famous for health inequality work because of Michael Marmot's work on employment grade and mortality. And I'd gone through the questionnaire, obviously, and I saw that there was this question about car ownership. So I asked Geoffrey, I said, why did you include the question on car ownership? And he said, oh, well, we're interested in physical activity, and we thought the car owners would be doing less activity than the people who didn't own cars. And therefore, they would have higher coronary heart disease risk. But of course, it turns out car ownership was rather strongly related to income. And we got a lot of data from that period showing that was the case. So it was yet another socioeconomic marker, but in the opposite direction. The car owners did, in fact, have somewhat higher BMI than the non-car owners, suggesting that Geoffrey was probably right. It probably did relate, in those days, to physical activity. But what we showed was that you could stratify by car ownership as well as employment grade, and you've got even bigger differences. You've got 4-fold differences in all-cause mortality between these groups. But the other thing we noticed in those analyses was that with car ownership, when you adjusted it for everything, for blood pressure, for body mass index, for smoking, for health status, etc., car ownership remained strongly protective, statistically, very, very robustly protective against coronary heart disease. This stuck with me, as you will see, for a long period. Then in 1989, I was lucky enough to get accepted on the advanced course on cardiovascular disease epidemiology in Courson-Dunk Priory, where I met Andrew Phillips, who is here somewhere. He's aged much better than me, so he'll be easier to identify. And Andrew and I were very interested in measurement error. And in fact, we did some skits about measurement error and meta-analysis and other things on the last day of the course. And in particular, we were interested in relation to HDL, which, as I say, the Caffili study had been set up about. These are the beers in Courson-Dunk, 9% proof, the one on the right. But more interesting fluids are the ones on the left. More interesting fluids are the ones on the right, which are your centrifuged samples. And when collecting these in Wales, often especially in early morning samples, etc., a few of them would be cloudy, which got me quite interested in triglycerides. And you'd see that triglycerides are hugely variable. They vary after meals, they vary after drinking large amounts of alcohol. The British Regional Heart Study, which Andrew was working with, had done an experiment where they fed people full English breakfast. And you saw the triglyceride levels zunk up. HDL cholesterol just stayed completely flat. So we thought, well, you know, one measurement, a single measurement of HDL cholesterol is going to be obviously more stable than a single measurement of triglycerides, which just have huge amounts of measurement error. And we wrote a series of papers with a load of simulations, all of which Andrew did, about this issue and about the measurement error, both in the exposure and in potential confounders. And then with triglycerides and HDL in the regional heart study, we showed that HDL appeared to protect against coronary heart disease. The blue lines are just the unadjusted, and then the red are the mutually adjusted. So the adverse triglyceride effect got substantially lower, but the HDL cholesterol effect was stable. But then if you modeled, I just flipped them so they all point in the same direction. But if you modeled the degree of measurement error in those variables, you could just change them by just changing the interclass correlation. And you could just flip the results. You could just make it, it's very simple to get it that the HDL cholesterol effect attenuated substantially and the triglyceride effect was robust if you took into account very considerably greater measurement in precision in triglycerides. Now, those papers had absolutely no impact whatsoever, but they did change both my career and Andrew's career. Andrew went off to do really useful work on HIV. I was a bit more gadfly-like, but I did start working on childhood diarrhea in Nicaragua and sexually transmitted diseases and HIV, and other things where measurement error wasn't the point, cause wasn't the point, because you know what the causes was. It was sort of doing something about them. And I was registered to do a PhD at UCL on how fibrinogen mediated between job stress and coronary heart disease. And I gave up the PhD because with these sorts of models, it was very clear there was no way the sort of study that I was proposing to do as a PhD would be able to say anything useful. It would just reflect measurement characteristics. So that was then the thinking about other ways. How can we actually get closer to cause? And I was interested in a method which only now, from 2010, was sort of renamed negative controls, but it had been used in epidemiology since Percy Stokes and Major Greenwood in the 1920s and 1940s, which was looking at factors which could not plausibly be influenced by an exposure. And if the exposure related to those factors, then you would worry about there being any causal relationship. And so this paper in The Lancet, the title being Smoking as Independent Risk Factor for Suicide, Illustration of an Artifact from Observational Epidemiology, we showed that there was a lot of literature about smoking and suicide in those days, showing quite strong observational associations. But it's quite obvious that the sort of confounders, social position, risk-taking behaviour, but also reverse causation, which is a form of confounding, such as people self-medicating with cigarettes, depression making it more difficult to give up smoking, etc. So we wondered whether this association really was causal. And these were data from a multiple risk factor intervention trial in the US. So we had the obvious negative control, which is being murdered. Because it's very unlikely that smoking can actually lead to you being murdered. But sure enough, there is the same association as with murder, being murdered, as there was with suicide. And now, unless the paramilitary wing of the Health Promotion Agency had started shooting smokers as the ultimate deterrent, it was not really plausible to have a causal analysis of that situation. Now, Matthias, who's here, and I then looked at other studies, and you just show that this is very replicable. Just to show that very replicable associations, when they're generated by the same biases, doesn't tell you anything about causation. The replicability, once you get to large-size studies, and many of them in the same settings, is not very informative. So now, I mean, this is, as I say, the first meta-analysis on a weighted average that was done on this. Andrew and I carried on writing these papers, and we used the examples of like smoking and suicide, smoking and depression, and smoking and cervical cancer, which was then of considerable interest, and saying if there was an infection which underlay cervical cancer, which wasn't then clear, which had a very large effect, it could easily generate the sorts of associations you see with smoking and cervical cancer. And we discussed that with depression. And I'd gone to an exhibition at the Photographer's Gallery of Bill Brandt's photographs. And I'd seen this wonderful photo by Brandt, which is in the public bar at Charlie Brown's in Limehouse. And the guy on the left, as you can see, he's looking a bit miserable, and he's smoking. And if you look closely, you'll see the woman on the right is also smoking. So I sent this in and I said, you just put this in as an illustration in this paper. And then the paper appeared, and I opened the BMJ, and I realized that the minds of BMJ editors are very different to my mind. Because they were too mean to pay for the rights for the Brandt photo, and this is the photo that they put in to illustrate this. So unless being bored to death gives you cervical cancer, it's difficult to see how this could be causal. Anyway, after the motorbike crash, I finally managed to write my MD, and we moved offices recently, and I found a copy of it. I hadn't looked at it for years, and this is in my thanks section. I say surviving fieldwork. Fieldwork can be fun, but it's generally hell. And an interesting sociological fact was that successful epidemiologists do not perform any sustained periods of field work. I was on the way to becoming a super tech. But I was doing them field work in the Whitehall 2 study, 1,300 blood pressure reactivity measures. Each one took half an hour to see if blood pressure reaction to a psychological stressor predicted future hypertension and coronary heart disease. The answer is no. That wasn't great either in terms of finding anything. So you probably think I'm going to now talk about how correlation does not imply causation. But I'm not, because correlation does imply causation. It's just that the causation is not the straightforward causation you might think of it as between the two factors. If something is replicably correlated, like smoking and suicide, there must be some factors which are generating that, which, if you understood those factors, would tell you something very interesting about those conditions. So so replicable findings are interesting, but you've got to work out what the causation is. So, I mean, an obvious example, the joke example is always, smoking causes yellow fingers and it causes lung cancer. So that's causal. That's what the causes are. The association between yellow fingers and lung cancer isn't causal, but it tells you about underlying causal factor, which if you identified it, must be important. Now, confounding is easy to understand, probably less well recognized, although it's becoming increasingly so, is a second form of bias, which is called collider bias. But just to say, if scrubbing your fingers isn't going to reduce your lung cancer risk, but there's adverts for how to do it on the web. So conditioning on common effects can induce associations, known as collider bias. So let's have an example here. Okay, there's a lot of epidemiologists here. Let's say there's two routes into epidemiology. One is willingness to do field work. The second one is being good at statistics. Now, that's not me. But as we all know, people who are good at statistics tend to be rather poor at maintaining eye contact. Okay. So we imagine that in the general population, there is no association at all between willingness to do field work and being able to maintain eye contact. But once you get a group of epidemiologists, you find one of them. If they're looking at your shoes, they are not willing to do field work. In fact, you get a minus 0.66 correlation between willingness to do field work and maintaining eye contact. And over the warm Chardonnay later, you can try this out on people. But obviously, it is only an on average effect. There's at least one statistician here who's very good at maintaining eye contact. So those are the two ways that you can generate replicable associations if you're stratifying on something which is caused in the same way by factors, or more likely is the straightforward confounding. But lots of things were, of course, coming out which showed about the same sorts of effect sizes as car ownership. You know, this 0.65 relative risk after adjustment for all the confounding factors. Just like car ownership, clearly not causal. You know, and there's two papers appeared back to back in two studies of health professionals in the US and the New England Journal of Medicine. They showed 35% lower risk of coronary heart disease from people taking vitamin E supplements observationally. They adjusted for everything, just like epidemiologists do, but those remained robust. Those studies were reported in the New York Times. Vitamin E greatly reduces risk of heart disease, studies suggest. Greatly reduced is a causal claim. Now, these studies were amongst health professionals, and they started taking a lot of vitamin E because, you know, they'd heard the news. Vitamin E was reducing the risk of heart disease, and at one stage in the US population overall, nearly 50% of people were taking vitamin E supplements containing vitamin E. Randomized trials came out. You can guess the answer. The large-scale randomized trials, up to 13 years follow-up, showed absolutely no effect of vitamin E supplementation on coronary heart disease. And, you know, there were clever people in these studies, and they stopped taking the vitamin E supplements when the randomized controlled trials came out. And I looked to see whether I could get. I thought I want to do negative control analyses on this. I want to look at accidents and violence as outcomes, you know, associated with vitamin E supplement use. So I started writing, in those days you write letters, to see if I could find the data. I could actually would be allowed to look at the data. And you can imagine the response in 1993 to such letters. It was not that polite. Well, often you actually got no reply. When you did get replies, they weren't that polite. I certainly was not going to get any possibility of looking at the data. And so I wrote this editorial for the BMJ on increasing the accessibility of data, saying that data should be a requirement of publication to make the data available. And the then editor of the BMJ actually was quite supportive of this, and he took it to his sort of editorial board and proposed it to them. And it got one vote, which was his. No one else was supportive of it. And I looked back at this again for preparing for this talk, and it was rather touching to see this. I say, if papers submitted to journals had to be accompanied by a disk copy of the data, but the great thing is it would have reduced submissions. There's too many papers. It would have reduced submissions. The same, when the vitamin E findings first appeared, it was in a conference presentation. And they all, in that conference presentation, they showed exactly the same association with beta-carotene. Now, the beta-carotene papers were never published in the New England Journal of Medicine or anywhere, but the news got out from the conference, and the people started taking beta-carotene. But you notice that that fell from 1994, and that was because 1994 was when the first very large-scale randomized trial of beta-carotene came out with very disappointing results. Now, after 1994, there was a tsunami of these things that came out. These were large-scale randomized trials costing huge amounts of money on all these things, including HDL cholesterol, the good cholesterol that I discussed, with just null results. Hormone replacement therapy was supposed to be protective against coronary heart disease. One after the other, these came out. Selenium and prostate cancer, vitamin D, and many, many things, and many, many others. And the remarkable thing is, this period, as an epidemiologist, this was had quite an effect. But the remarkable thing is, this is just forgotten. That these things happened. No one has gone back to revisit any of these things to say why did they get the wrong answer. These were very replicable associations, but they just weren't. What one thought was the cause wasn't the cause. And of course, today's thing which causes everything is ultra-processed food. And here's a paper about ultra-processed food and suicidal ideation. Well, it's a sort of negative control study in my view. It's just people aren't treating it as a negative control study. They should get homicide, I guess. Anyway, as I say, I went off to do other things. I was doing work on HIV and STDs, and Matthias led a trial with many other people in Nicaragua, which is a very large-scale randomized controlled trial of condom promotion. This was doing trials in situations when people said it was just not possible to do the trial. This was a cluster randomized factorial trial of either leaving condoms in rooms or handing them out to people using motels in Nicaragua or no condom. And the other factor was health promotion, was putting up posters telling people to use condoms in the rooms or no poster. What was found was that amongst commercial encounters, there was quite a large effect of promoting the condom, providing condoms had a large effect on condom use, which was verified by retrieving the used condoms from the rooms. I did not do the field work on this occasion. And the health promotion had, if anything, an adverse effect on condom use. And the other one was looking at actually what happened in sexually transmitted disease consultations in India, where it was promoted that the doctors should be telling people to use condoms, telling men to use condoms. If they go for an appointment, they're obviously at risk, and therefore they should be using condoms. And we interviewed a lot of doctors. We got a lot of medical students to sit in and watch doctors while they were seeing patients. And in the interviews, and when they were observed, knowingly they were being observed, large percentage of them were saying that they were giving out condoms. We then got a group of people to go into the STD clinics as secret shoppers. Not one of them was told to use condoms. Those sorts of methods of getting data, interviewing doctors, etc., are worthless, are often just worthless. Other observational studies were carrying on with health inequalities work. This was with Danny, who's here. This was in 1996 in the BMJ. We looked at Tory voting Conservative, and mortality. And we showed a huge protective effect of voting Tory. It's just incredible. The higher the Tory voting, the lower the mortality. Now, the fact is the Tories should have said, vote Conservative, live longer. But because we'd framed it in this I'm all right, John way, saying that it's people who didn't require the use of state services, state health services, education services, didn't require welfare and employment benefits, etc., would vote for the party that systematically would take those apart. That the Conservative Party called for the editor of the BMJ to be sacked and said that I was a well-known Marxist. Now, I'm going to tell you something that is true. I am not now, nor have I ever been, well-known. Anyway, Patrick mentioned Mendelian randomization. I haven't got very long, so I can't. But Mendelian randomization is based on the magic of meiosis. Is that if you actually have within family data from parents to offspring, it truly is randomized data. And if you group people by their genetic variation, not by their phenotype, if that genetic variation influences a phenotype, then you get two groups which are balanced by confounding factors and for which reverse causation is not possible. So analysing by genotype is like an intention to treat analysis in a randomized controlled trial. And here I mentioned selenium, and here's a MR study looking at circulating selenium levels and prostate cancer. And in the randomized controlled trial, there was no benefit. And in the Mendelian randomization study, suggested no benefit, despite observational studies strongly suggesting selenium was protective against prostate cancer. Now, there's a huge number of caveats for Mendelian randomization. If you've got an hour later over the warm Chardonnay, I'm very happy to talk about them. I'm just going to give one more example, which is C-reactive protein, which is an acute phase reactant. It predicts coronary heart disease very strongly. When you adjust for all the confounders, it still predicts coronary heart disease. Everything bad is related to CRP. Atherosclerosis is inflammatory, puts your CRP up. So many factors that do influence risk of coronary heart disease relate in the right way, if you like, higher CRP, higher risk. The genetic variant is not related to any of the confounding factors. Mendelian randomization, in this case, with a sensible variant, does what it says on the tin. And of course, when the MR studies were done, there was no evidence of protection. And the pharmaceutical industry were trying to develop CRP inhibitors at this time because it was thought to be the new cholesterol. But they stopped on the basis of MR results. But the thing to look at here is that, again, look at that. Look at the result at the top. That is what a totally confounded association looks like after adjusting for as many factors as you can. These are the sorts of things that get reported and taken seriously. It was a good job I gave at my PhD because fibrinogen doesn't appear to be causally related to coronary heart disease. HDL cholesterol I've already mentioned. There's now terrible Mendelian randomization studies. They're really awful. Anyone who does reviewing or editing knows that. This is literally an exponential showing the rise of MR papers. In about 4 years' time, every paper in the world will be a Mendelian randomization paper if this continues. But it can be extremely useful. And genetics can also be useful in trying to learn why things were confounded. So with HDL, we've looked at sepsis death because lipoproteins very strongly relate to sepsis mortality. And observationally, small HDL relates. But interleukin-6, which influences HDL cholesterol, which lowers it, and which is the target of tocilizumab, which reduced mortality in COVID, of course, which Mendelian randomization, by the way, showed before the trials appeared. IL-6 is a potential strong confounder of this. In fact, that explains about 50% of the observed association. But this does actually suggest that IL-6 receptor blockade might be useful in sepsis. Patrick also mentioned triangulation of evidence, which is just using many different forms of evidence, all the different methods, of which there are many, which can strengthen causal inference to actually come to a conclusion. And these formal ways of doing that are being developed. But I'm going to finish by talking about between population causes. Because when thinking about what really generates, we know things that are influencing health, things that lead to large-scale differences between populations are what's important. This is John Higginson back in 1960 saying that these huge differences are going to tell us about causes. And Richard Peto, who's here, and Richard Doll wrote this brilliant report in 1981, pointing out that if something is changing over time, if migrant studies show if people move from one place to another in either 1 or 2 generations, the risk changes substantially. And geographical differences tell us that in principle, these diseases must be preventable, cancer in this case. But only 75 to 80% of the conditions are in principle preventable, but only about a third was their cause known and were they actually preventable. And based on this, then Richard Doll said absolutely reasonably for the time that in 1 or 2 decades we should identify the other causes because smoking had been, of course, identified. Paul Brennan, who's also here, and I updated these analyses a couple of years ago, and still 80% are preventable in principle, and the amount that could be prevented by what's known is still the same. That's despite HPV and cervical cancer being identified, etc. And the reason for that is because smoking has fallen, a lower proportion. You can actually say what the cause is because smoking has fallen. But the preventable in principle is very large, and this is what epidemiologists should be targeting. And Jeffrey Rose, in sick individuals and sick populations, as I say, if you read one epidemiology paper, read this one. He suggested this thought experiment about population in which everyone smoked exactly 20 cigarettes a day. So let's imagine that everyone in Bristol, the town I live in, has to smoke 20 cigarettes a day. When you wake up in the morning, you're given your pack of Capstan full strength, and you're not allowed to go to bed till you've got through the 20. And Bristol's edgy, so people probably would smoke. And in Bath, no one smokes at all. The dictators have come in, and Bath's posh, so yeah, people won't smoke. And then you follow them up for 50 years, and obviously Bristol would have a vastly higher lung cancer rate. But within Bristol, what causes How does smoking relate to lung cancer? Well, it doesn't, because everyone's smoking exactly 20 cigarettes a day. Smoking doesn't relate to lung cancer. What causes one individual rather than another to get lung cancer? Well, there's genetics, and lung cancer is heritable, although it's quite low heritability, and a chunk of that heritability is through the heritability of smoking behaviour. And between Bristol and Bath, what causes the huge difference? Of course, it's smoking. So the causes of between individual and between population health differences can be entirely distinct. So let's think about, about Winnie. Okay, here's Winnie. By the way, this is the only paper I've ever written which is worth reading. It's never cited. Please, cite it sometimes. Read it. This is Winnie. She's 100. She's been smoking for 93 years, and she ain't quitting now. Everyone thinks, well, let's study lots and lots of Winnies, and we'll discover, how to resist lung cancer. But let's think, Winnie, when she was 60, it's her 60th birthday. There's a knock on the door. It's 11. She goes and opens the door. It's the postman with her cards. Gust of air. She coughs up a metaplastic lesion. And Winnie then makes it to 100. Now, Winnie was just lucky. But if Winnie hadn't gone to the door, there'd be some other person who would have been lucky. The counterfactual would have been that they were lucky. Between individuals, chance, luck, things like stochastic mutations, stochastic epigenetic changes, etc., are likely to explain a chunk of the difference, and they're not targetable, sensibly targetable by any intervention. Stephen Frankel is here, and I and Charlie Davison were involved in studies in the late 1980s about what people thought caused heart disease in South Wales, the ethnographic studies. My hypothesis was you're going around Wales, post the miners' strike, socialist countries it was then, and people would say, if you say, what causes lung cancer, coronary heart disease, they'd say Margaret Thatcher and capitalism. That's possibly, that was a I was sort of hoping for that. What they did say was genes and chance. If only I'd listened to the punters at the time, and because at the individual level, when all you're perceiving is individuals, the people you know, everyone knows an Uncle Norman or a Winnie who've survived a long time, and even more, I think most remarkably, is when you look within one individual, you get incredible evidence about how. About the stochastic elements. You consider someone gets a cancer of one kidney, and you think of all the causes of kidney cancer, then their other kidney is perfectly matched on any of the exposures we think of, from germline, genome, any modifiable exposures, germline, genome, smoking, etc. They're perfectly matched, and yet, when someone develops a cancer of one kidney, their risk of getting cancer in the other kidney is about 40%, relative risk 1.4. So about the same sort of things as car ownership, compared to just a random kidney in that population. It really is remarkably small, as Richard Dahl had pointed out. So, but I say Richard Dahl pointed this out in relation to breast cancer. The data of breast cancer, there's a huge amount of data on breast cancer, so it shows this 1.4 sort of effect overall. At younger ages, it's much more, but of course, many less of the cancers occur at the younger ages, like Rory pointed out about relative and absolute differences. When you look across all ages, it's 1.4. So imagine you have your two populations, the most extreme differences in the world. For kidney cancer risk, one has 5-fold the risk in another population. There's something in the environment which leads to 5-fold the risk of the kidney, of kidney cancer. And what happens is, in the environment which has 5 times the risk of the kidney cancer, all the other kidneys in that population have that 5-fold relative risk, and therefore your other kidney has a relative risk of 1.4, as only compared to the other kidneys in that population, and the same as you'd see in the low risk population. If anyone has got an explanation for this, which isn't in terms of some population level exposure, then Paul Brennan is here. Put your hand up, Paul. Come and tell Paul or I what the explanation is. So ubiquitous exposures are exposures which tend to be similar for people within geographic regions, and for which the variation between individuals is low. Measurement error becomes a major contributor to between individual exposure differences. Chance together with individual susceptibility, not measured exposure, discriminate who gets sick, e.g. who gets lung cancer when everyone smokes 20 cigarettes a day. And Paul and others have done some work, for example, on kidney cancer using mutation signatures, where in Japan they find a mutation signature which just isn't seen, is only seen at very low levels elsewhere, suggesting there is an environmental factor which is ubiquitous in that environment that isn't seen or is seen in very low levels in other environments, which is the sort of clue you need to try and hunt down the environmental causes that are potentially manipulable that generate these conditions. And I'm going to finish on my favourite piece of work because it took so long. And this is milk as ubiquitous exposure millennia ago. And when Shah's in the room too, when Shah and I wrote our second paper on Mendelian randomization, we were very excited that we could study milk using Mendelian randomization because everyone had heard of lactose intolerance. And the story we'd learnt at university and is still taught is that lactose intolerance led to, there was a change when milk arrived. There was people couldn't drink milk because everyone was lactose intolerant. The ancestral allele is lactose intolerant, and therefore, a mutation which led to being able to tolerate milk, drink milk, just took off. That was the story, and that was why it's the most rapid, the most rapid evolution in humans. So we went and the variant had been discovered in 2002 in Europe. We genotyped the study that Shah had set up, and Deborah Lawler, who's here, did the field work for. Shah did some of the field work. We genotyped this study for the lactose persistence genetic variant, and it hardly related to milk consumption. Now, we couldn't get this published because people said this just can't be true, but it was the biggest study at the time. It got bounced from journal to journal. And we also tried to suggest that the standard evolutionary story was unlikely to be true and proposed actually the thing which we now think is the case. But now this work with with multidisciplinary team, Richard Evershed, who works on ancient pottery to say when milk starts to be used, and Mark Thomas, who works on ancient DNA as a geneticist, human geneticist. We worked using UK biobank data and pottery shards and ancient DNA data and a lot of environment data, paleoarchaeological data about populations to come up with the notion that milk consumption became just increasingly widespread from 10,000 years ago. Lactase persistence was virtually unknown till 3,000 years ago. For 7,000 years, people were happily drinking milk. People, all of them were drinking milk. Because lactase persistence hardly relates to milk consumption. The increase in lactose persistence genetic variants were only seen from 3,000 years ago. Very weak association between lactose persistence genetic variation and milk consumption in UKB, like every other study. Increasing lactose persistence temporally and geographically was related to indicators of population size and density. Basically, the hypothesis is that when populations got large enough and dense enough, that gastroenteritis causing bugs would be maintained in those populations. Then the selection occurred because if you're drinking milk and you're lactase non-persistent, you get a bolus of lactose in the colon, and when you get gastroenteritis, you're more likely to dehydrate and die. And you'll die before you're reproductively active. It's kids who will be dying. And so we think the lactose persistence, like virtually every form of rapid selection, is actually due to infection. So that's the hypothesis. Milk consumption 10,000 years ago was a ubiquitous exposure. So my summary is within population studies identify individual level causes. Between population studies can identify causes generating major international differences in disease risk. These within and between population studies relationships, depending on what the specific factor you're looking at, particularly disease and particular exposure, they may be the same or different in the two different contexts, but it's worth learning whether they're the same or different. Exposures being ubiquitous within populations may be one reason why identifying environmental causes of disease, which are clearly predominant, has decelerated, and new methods are required to address this. Infection is a potential example of a ubiquitous exposure, with age at initial infection influencing outcome. This is what I'm going to spend the remainder of my career working on. And their implications of chance as a major factor for individual risk within populations has implications for personalised medicine. Because if chance is a large difference, the idea of actually personalising things is obviously somewhat diluted by that. I mean, prediction you can do well, because prediction is when it's through things which are actually early indicators of disease, or not that early indicators of disease, you can do pretty well, but actually on causes, not so much. So, for example, with statins, which we've heard a lot about, you reduce outcomes by 25% with a statin, and there's so much written about let's personalise, let's personalise the use, let's sort of find out. You see these people writing that if we could only find out who are the small percentage of the population who are actually saved by statins, we can save money and we can reduce the burden of tablet taking. You can't know in advance which that group is. It is like, but with statins, every day if you have to pull the trigger, we take statins, you have one bullet of the 4 removed, you're 25% reduction in risk. It's probabilistic though. And I feel quite strongly about this because I am a Winnie. When I went through that, when I went into that car, I was not wearing a helmet. Helmets for motorbikes were quite difficult to get in Nicaragua at the time, and I arched beautifully through the air, turned round and went with my back against the seat next to the driver. I made a perfect arch, a rather improbable perfect arch. If I had been wearing a helmet, my head would have been a lot heavier. I would not have made that perfect arch. In retrospect, so I was lucky that I was not wearing a helmet. But you can only, but on average, wear a helmet. Caroline and I were in Bangalore recently, and we saw this sign: wear a helmet and be safe. So, and that's true. That's all you can do is work on. You can't predict backwards, sadly. You can't know. So I definitely am a Winnie. In my MRC timeline, I say I was very lucky enough to be in the MRC unit South Wales. 1995 onwards, I've been PI or co-I or sponsor to 54 funded MRC applications. I had to change this slide yesterday because it was 53 yesterday. But yesterday I heard a fellowship had been awarded for studying the lipids and the HDL lipids and sepsis story further. So I'm really pleased about that. I was then, MRC Unit South Wales closed. All its materials were transferred to social medicine in Bristol. In 2001, the first MRC Welcome Core Support for ELSPAC. I was then director of ELSPAC 2005 to 2017. 2008 to 2013, I was director of the MRC Centre for Causal Analyses and Translational Epidemiology. We were told we had to have a title with translational in it, and that was the best we'd come up with. We stupidly didn't Google it, because in Ireland, Kate means knackered, worn out, etc. But luckily, we became a unit in 2013, the Integrative Epidemiology Unit, the IEU. But as I say, 54 funded MRC grants, but you want to see the number of failed grants I've put in. But the truth is that some of those failed grants, looking back at them, it was really lucky they were turned down, because, of course, you never feel that at the time, but I would have spent time, it would have been like the fibrinogen and job stress type thing. I would have wasted my time on things so I was lucky. Some of the grants, it was good news, they weren't funded. I've mentioned the people who are here. I've worked with so many fantastic colleagues, nationally, internationally, over many years. It would take the 40 minutes to read out the names, but most people who know me will know the person has to be thanked is Julia McKay. Who's worked with me, who's put up with me for 25 years as my PA. Thank you.